welcome back. In lecture 52, I will be discussing about DNA sequencing. During the last lecture, I was discussing about agarose gel electrophoresis and polymerase chain reaction. But through polymerase chain reaction, we can increase the amount of DNA, but through DNA sequencing, we can establish the exact sequences of nucleotide present in some particular DNA segment. So, particularly I will be discussing about principle of DNA sequencing, then the sequencing reaction, followed by like new development, I would say automated DNA sequencing method. So, now here, before starting the DNA sequencing method, I would like to mention that sequencing means the process by which you determine the exact order of the nucleotides in a given region of DNA. So, we do DNA sequencing to decode the genetic information present in DNA. And how we can carry out this reaction, how we will determine the sequence, that I am going to discuss in more details in the next slide. And as you can see, the scientist is very famous, Frederick Sanger. So, he received Nobel Prize twice. Only I believe out of 5 people across the world who received Nobel Prize twice, he is one of them. And again, he received Nobel Prize in same category. Both the time he received Nobel Prize in chemistry. And for DNA sequencing, he received Nobel Prize in 1980. So, this is for DNA sequencing. Then what about 1958? So, he also developed some technique to sequence protein. So, he determined the sequence of one protein called insulin. So, for that also he received Nobel Prize in 1958. So, this is just interesting, that is why I just mentioned. And now, let discuss about DNA sequencing method. This is a DNA sequence. For example, if I write here 3' A, T, G, C, G, T, A, G, A, T, G, C, T, A, T, G, C, A, G, just some sequence here. Now, I am writing this sequence so that I will be able to explain it, but our goal will be to determine the sequence of this stretch of DNA. Without writing the sequence, I cannot explain it. That is the reason I am writing here. So, now this is just one strand of the DNA. For the DNA sequencing, we have to just sequence one strand of DNA. As you know that DNA strands are complementary, so that does not matter. If you just decode the sequence of one strand, that is good enough for us. So, now if this is the DNA sequence, then we can use some primer. Say for example, 5' T, A, C, G, C, A, T, C. Say this is a primer, 5' to 3'. So, this primer can grow. So, this is primer. Now, maybe we are not aware about this sequence, as I already mentioned. So, we have to determine the sequence of that segment. So, how we can do this? So, this primer will bind to this template sequence, and then if we use DNA polymerase, then this primer will be extended. So, whatever, just like PCR, just like replication, we have to carry out this reaction, and then we will add some additional trick and strategy so that we can decode the sequence here. So, that is why I initially explain about replication and then I explain about polymerase chain reaction so that it will be easier to understand about DNA sequencing here also. So as a result of that, in this tube, if we are carrying out the reaction, then in this tube you should have DNA polymerase, you should have this DNA template, whatever the sequence I mentioned, you should have primer also, but only thing here, you just need one primer or just one primer sequence. You don't need two primers like polymerase chain reaction. Here we are like trying to determine the sequence of one strand, that's why. So DNA polymerase will start adding nucleotide. So then this primer will be extended. So now, so what will be the next nucleotide here? So it will be T. T should come here. Then A, then C, G. It will go like this. But if it is going like this, then again, like just like polymerase chain reaction, we are just synthesizing the whole DNA. But that is not our goal. We need to find out the sequences of this region. We have to determine the sequence. So as a result of that, I am just erasing this part. So this is not our goal. What we are going to do is, we will add some additional reagent in this tube, some kind of, I would say defective nucleotides, some kind of problematic nucleotides there, so that whenever those nucleotides will be incorporated during the chain elongation, then the chain will not be extended anymore, it will stop. So that is the strategy. So as a result of that, I would say, if in this tube, for example, this is my tube number 1, in this tube, in addition to all the reagent required for this chain synthesis, the DNA polymerase, magnesium ion, all DNTPs, everything there, right? So I have DNTPs, polymerase, then template, primer, everything there. In addition to that, I have some defective T. So some additional nucleotides, I am saying T star, that is the defective T. So I have all these 4 nucleotides ATGC, but in addition to that, I added another nucleotide in this tube, that is some defective T, some problematic T here. Whatever happening here, I am trying to explain, this is happening inside this tube. So then the next nucleotide will be added here, the T. Now we have two options. One option is that the good T, the normal T which is present in this DNTP mixture, it can be incorporated here, or this defective T also can be incorporated here. If the normal T has been added here, then the chain will grow. But in case you have this defective T, the T stars get incorporated, so then you will have the T star. So it will not grow anymore, it will stop there because of this defect. Then in this tube, I have many primers. So as a result of that, if I take another primer here, T, A, C, G, C, A, T, C, and then this good T or the normal T incorporated. So as a result of that, this chain will be extended. So then next nucleotide should be A, C, G, then again A, then again another T. So maybe this T is the defective T. So as a result of that, again it will stop here. Similarly, it will not grow anymore. So another primer, 5 prime. So I am not writing the primer sequence anymore. So here this is the primer, and then again T, A, C, G, A, normal T incorporated here. So as a result of that, again it will be extended A, C, G, then again the bad T or the defective T, so it will stop here. So then the idea here is, in this reaction, in this tube 1, I have every component. In addition to that, I have this defective T. So as a result of that, whenever this defective T is getting incorporated, then the reaction stops. That chain cannot be extended anymore. Then as a result of that, what I am going to get it, like in this tube, after the reaction we will get different length product, and all those product will be end with the defective T at their 3' end. So this is the result from this tube 1. But here we have to be careful that the concentration of defective T should be very, very less. Generally, the ratio of this defective T should be almost, I would say, 100:1. 100 times it should be the normal nucleotide, normal T, and only 100:1 ratio, only 1% should be this defective T. So that at the beginning only defective T will be incorporated, then the reaction will stop, it will not be proceeded further. But that is not our goal. Our goal is to try to carry out the reaction as long as possible, so that we will get different length product, and all of those products are done with T at the 3' end. Now the thing is, I am just mentioning this tube 1 here. What will happen then if I have 4 tubes, like tube 1 already I mentioned, and now tube 2, tube 3, tube 4. And in this tube 1, we have defective T. In tube 2, for example, defective A, tube 3, defective G, and tube 4, defective C. So as a result of that, in this tube, in tube 2, we will have different length product. This is 5' to 3' direction, and everywhere at the end, what nucleotide should be there? It should be defective A at the end, right? Because I have defective A in this tube 2. So as a result of that, now if we try to understand what is happening in all these 4 tubes, we will get different length product, all the possible length product in these 4 tubes. And now what will happen if we run a gel here? If we run a gel, this gel is little bit different from agarose gel electrophoresis, because our goal here is to separate DNA molecule if they are just differing by one nucleotide. Because as you can see, like this DNA molecule here and the next one, they are differing by just 4 nucleotides. Similarly, here this one, you have just one extra residue of defective A compared to this DNA strand, because after this in this template you have T, so that's why this is the smallest product in tube number 2. So as a result of that, our goal is to separate these DNA, this DNA of different length, they are differing just by one nucleotide. So now we will be loading our DNA sample here in this gel. This gel is generally made by polyacrylamide, not by agarose gel, and some additional parameter we have to incorporate it in order to separate those DNA properly, so that we can even see just one nucleotide difference. Now the thing is, if we load here the tube number sample from tube number 1, tube number 2. So sample from all these 4 tubes we have loaded in this gel. Similarly, it will be electrophores from negative to positive direction. Now, in which tube we have the smallest product? Based on the template, if you see, actually we have the primary same in every tube, right? So we have the smallest product in tube number 1. As you can see, it was A here, and that's why this is the defective T. So in tube 1, we have the smallest product. So as a result of that, after this gel run, we will be seeing the smallest product here. And then the next one, you have T, so that it should be defective A. So you will be seeing something like this here. And then C, so defective C is in line 4. So here, then here. So I am not following whatever mentioned in the sequence in the template, but if I just put randomly, then we will be getting something like this. So as a result of that, if this is the gel, then you can determine the sequence. From here, you will get the smallest product, it migrated maximum. So this is the first nucleotide. So what is the first nucleotide? It should be the it should be the T, because defective T was there in the tube 1. So the sequence should be 5 prime T. Then so you have here defective T, A, G and C. So the sequence will be T, A, here this one, C, G, A, T, C is going on like this. So that way, from the gel itself, you will determine the sequence. Now here the question is, what was the defect? I just mentioned that it's defective T, defective A, defective G. What was the defect? The defect was here nothing but a small change in the nucleotide. So as you know that this is the pentose sugar present in DNA, right? So this is the 1 prime, 2 prime carbon, 3 prime, 4 prime and 5 prime carbon. Now in DNA you have OH here. That means in deoxyribonucleotides you have OH at the 3 prime end. This defect was a very small change. Instead of OH, you have just H. So if you have H, then what will happen? Whenever this nucleotide will be incorporated, then it will not be extended, because I mentioned before during the replication that this OH, it helps for the nucleophilic attack, so that the next nucleotide can be added. So as a result of that, if you replace this OH with H, then we would say this is di-deoxyribonucleotide triphosphate. I am not showing the phosphate part here. So di-deoxy, because you don't have oxygen here at the 2 prime position, because this is the DNTP analog, but here also you don't have the OH. That's why di-deoxy, both the oxygen atoms are missing here. So that's why we mention this as DDNTPs, di-deoxyribonucleotide triphosphate. So as a result of that, you can understand that defective T means you have the di-deoxy this thymidine. So as a result of that, whenever it will be incorporated, it will not be extended anymore. This is the trick, and this was developed by Frederick Sanger, and that's why he received the Nobel Prize. So he discovered this principle and this method. So now, if you see here, whatever I tried to explain, here also you will get almost similar thing. So as you can see, this is the DNA, and which need to be sequenced, and this is the sequence again we are mentioning here, but after the reaction you will get the sequence, but just for explanation it has been shown. Now you have this primer, and this primer will be extended in this direction. So now here you have this tube number 1, 2, 3 and 4, and in tube 1 you have this dideoxy ATP and dideoxy CTP here, in tube 3 dideoxy GTP and here dideoxy TTP. So as a result of that, whenever this reaction is happening, you will get this sort of product here. So you can see all this product, they are done with the G residue at the end, right? Similarly here T residue at the end. Now if you analyze through gel whatever I explained, you will be seeing something like this sequence. So here the migration from this to this direction, right? So as a result of that, this is the smallest product. So this is G, as you can see. So as a result of that, you will be getting this sequence from 5 prime G, A, because this is present here, A, then T, T. So the same sequence present here. So that way you will get the sequence. So this is the way how we determine the sequence of a particular segment of DNA. Now in this method, some important features I should mention, at least a few of them I should mention. One is this amount of DNA is very low, what you can see here. But how I will observe that? So because of that, we use some kind of radioactive isotope. This primer here, this primer, this 5 prime end, this is labeled with some, P32. This is the radioactive phosphorus. It is labeled with that. So as a result of that, after this gel run, although we have very small amount of product in that gel, but you can do some additional experiments so that we can put some extra film and then we can develop that film, so that all this DNA molecule, they are like whatever you can see this band, they are getting extended from that primer, and each primer contain this radioactive atom, radioactive phosphorus. So as a result of that, they will because of this radioactivity, they will put some kind of impression on that extra film, and that is what you are getting here. So just to make this thing more sensitive, we add this radioactive phosphorus with the primer, and you can analyze this gel in this way. So this is all about dideoxy sequencing method. This is Sanger sequencing, commonly known as. And now little bit of modification of this technique is now very robust, and generally we say automated DNA sequencing. This is nothing but the same whatever I explained. The basic principle is same. The only thing is little modification. Instead of the radioactive thing, here we use some fluorophore. So we don't add radioactive phosphorus with the primer here. Rather, here this dideoxy ribonucleotide triphosphate, that DDNTP. So they are actually labeled with 4 different colors. For example, as you can see here, you have the dideoxy T is a color here mentioned, and dideoxy A, here also green color, dideoxy G, and here dideoxy C in blue color. Just 4 different fluorophore should be added with this defective ATGC. That's all. So if we do that, actually we can carry out this reaction in just one tube. There we are doing in 4 different tubes to analyze that in the gel. But here, since all these 4 nucleotides, these dideoxy nucleotides, they are labeled with different color, we can carry out in same tube. The reaction will be happening in the same tube. So after the reaction is done, so you will get all possible different length product in single tube itself. And then that product, whatever product here you get after the reaction, you have to load on a small or narrow capillary tube. This is called capillary gel electrophoresis. Capillary electrophoresis. So some matrix is present inside this capillary, and now again this DNA will be migrated from top to bottom in this direction. This is just like capillary electrophoresis, just the same principle like electrophoresis. So now again the smallest product, they will migrate faster, just like gel electrophoresis. So as a result of that, when these products are coming out from the capillary tube, here you have one detector and here you have the laser source. So the laser will go in this way and the detector will detect that which color is passing. So now during this separation in the capillary tube, as you can see, even the single nucleotide difference, those DNA can be separated, and we can plot that data in this way, this kind of curve. So they will give whatever this color is getting detected, so it will plot here. For example, this blue color peak here, consecutively 2 blue color peak. What does that mean? That means, because as I already mentioned here, the C, that dideoxy C here colored with this blue color fluorophore, for example. So that means here you have the C residues coming. So that way, continuously this DNA molecules coming out from the capillary tube, and the detector is detecting, and you are getting this kind of chromatogram and this kind of peak of different color, and from there you will get the sequence of the DNA present in the template itself. So but this is very powerful because as you can understand, this very efficient process, we can carry out this reaction in just one tube, and you don't need to use any radioactive thing and that kind of complicated gel analysis and all those things. But this is very efficient method, so we can sequence a larger fragment of DNA at a stretch, and that's all. And you can follow any of this textbook for this sequencing part. That's all. Thank you very much.